to present our last CU Talent webinar. This is Laura Martin. I'm reSEArch-EU project coordinator. And today we are presenting, our last CU Talent webinar. It has been almost 8 different webinars with different topics in which different researchers, early stage researchers, have been participating, sharing their research with the different partner universities of this alliance. Today is the time of the webinar entitled Environmental Pollution. And we have our speaker from the University of Western Brittany in France. Today we have the pleasure to have with us Daniele Mello. She's a postdoctoral researcher at the University of Western Brittany, as I said before, and she's also part of the Lemar laboratory, and she's going to give us a talk that is related with the project she's coordinating, which is marine risk assessment using oyster immune cell model to support coast environmental health. This project, if I'm not confused, is funded by the French government. Is that right, Daniele?
Yeah, it's a European initiative from the Marie Curie Action together with the Brittany region. So it's a co-funds program.
Yeah. Okay, thank you so much. And I find it very interesting. It's very close to the research I'm dealing with, and more, all what regards with immune system is taking protagonism, in the sense that once we look for responses of exposure and effects of chemicals in the environment, normally or frequently we have been focused in the metabolism of degradation of chemicals, and more it's important to take also into consideration how the immune system is being affected. So her talk today is entitled Exploring the Close Connection Between Metabolism and Immunity in Search for Immunometabolism Disrupting Chemicals Treating Marine Life. So thank you very much, Daniele. The floor is yours. Thank you.
Thank you so much, Maria. It's a real pleasure to be participating in this CU Talents program. I believe it's a great initiative for early career researchers such as me to be able to talk about our research and what we are passionate about. And this is a subject that I'm really excited, and I'm glad to be able to share with all of you. So you already introduced me. I'm a postdoctoral fellow at the Laboratory of Marine Science, Lemar, at the University of Western Brittany in France. And so as a, let me see here. Okay. As an immunotoxicological researcher, my research interests have been greatly motivated by the fact that, I always wondered what these toxic chemicals that have been drastically increasing worldwide are doing to our health and the health of life on our planet. And this is very serious because, chemicals, their production, it's increasing exponentially. And not only the amount that it's being produced, but also a great diversity of these chemicals. We have new classes of chemicals emerging each year. And so In regards to humans, we already know that they cause a great problem to our health. So this was out in 2017. So the Lancet Commission on Pollution and Health has summarized that each year we can have around 9 million premature deaths occurring worldwide due to pollution. And this is 3 times greater than death caused by AIDS, malaria, and tuberculosis combined, and 15 times greater than deaths caused by wars or other forms of violence each year. So it's a big issue that before this was published, like nobody had an idea of like the amount of the problem that this is taking in our human health. But what about these effects on marine life? So we have several researchers working on the effects of chemicals on marine life, but we still have no idea of the real impacts they are causing. And we know it's a big problem because all the chemicals that come from our activity, from human activity, we know that in one way or the other, they will end up in our oceans. And in the oceans, they can accumulate in the water, in the sediments, and inside the organism, and even more accumulated throughout the food web chain. So this is a serious problem. And together with this, we know that we have been experiencing massive disease and mortality events in many marine species. So here I bring just 2 examples. We have the Pacific oyster mortality syndrome, which has been a serious problem for aquaculture production. And also many of you may have heard of the coral white syndrome, so many corals dying. And for many of these diseases, we know some of the agents causing them, so the etiological agents. So we know that there are bacteria, viruses involved, also some abiotic factors like temperature. But I'm also convinced that pollutants may also play a major role in such mortality events. And why? Because we know that several pollutants can cause metabolic and also immunological effects on marine organisms. So historically, these effects have been addressed separately. Many researchers will focus on the effects of chemicals on metabolic parameters such as growth and development, while others will focus on immunological parameters and the susceptibility to pathogens. But as an immunotoxicological expert who had also a very strong training in mitochondrial metabolism during my previous postdoc in the United States, I would like to bring a more integrated vision to study the pathophysiology of such marine diseases. So this brings me to the field of immunometabolism. The term immunometabolism was coined very recently, so only in 2011. So it's a very novel field and refers to the metabolic process that regulates immune responses. So these regulation, they usually occur due to environmental cues. So here we have an inactive immune cells, and when we have such cues like pathogens or temperature, for example, we will have some metabolic shifts occurring within these cells and promoting the activation of these cells. And this is very well documented for vertebrate cells. So it's very well studied in the biomedical field, and it's involved in several diseases, but we know very little about such mechanisms in marine invertebrates. I have some very fresh results demonstrating that this could be a conserved mechanism also with marine invertebrates. So here, when I exposed oyster immune cells to poly-IC, which is a molecule that will mimic a viral infection. So we can see, so here are my control cells, which only have seawater. And then when we add a larger amount of poly-IC, we can see an immediate metabolic shift of these cells in terms of NADH production, which occurs throughout the Krebs cycles to promote ATP production in the mitochondria. So we can see this shift happening in a matter of minutes. And so this is a normal immunometabolic process that can occur for the activation of immune cells. But what I'm interested in is that many pollutants, they can perhaps act as some of these normal environmental cues and disturb this metabolic shift, potentially activating these cells, but not in a scenario that it's necessary for the organism. So this can lead to unnecessary inflammatory outcomes. And so such types of pollutants that can disturb this immunometabolic pathways, I am proposing a new category, which is immunometabolic disrupting chemicals. So it would be in the same lines of, you might have heard of endocrine disrupt chemicals, which have been a big deal in the past years that we know cause several health effects. And so now I'm proposing for us to study such chemicals that can cause immunometabolic disruption. And so I'm very interested in how these could be very important factors in the pathophysiology of diseases. So one of my main research interests is to identify which chemicals can act as immunometabolic disruptors and understand the outcomes of these exposures to the susceptibility of diseases in marine organisms. So this brings to my current postdoctoral project. So as we discussed earlier, this project is supported by the Marie Curie Action and the Bienvenue Co-Fund program. And it's under the supervision of Charlotte Corporeau from IFREMER, Stephanie Madec, and Claire Julio from the University of Western Brittany. And so the MAHIM project was developed under a holistic perspective that global changes are co-occurring and synergistically impacting our planet's health. So at the same time that we have climate change, which has been predicted to significantly impact the composition of the marine microbiota, potentially towards a higher abundance of opportunistic or pathogenic microorganisms. Together with this problem, we have this exponential rise in the production and use of syntactic pollutants that we were talking about. All of this can act synergistically to impact our coastal ecosystem. And to represent this coastal environment, bivalves are commonly used as sentinel of the quality of the environment. And so in my studies, I am mostly using the oyster Classocea gigas as a model to understand, how these can affect disease sociability and compromise human, animal, and also ecosystem health. And as a chemical of interest, the MAHIM project will focus on antiphylline compounds. So these compounds are they can also be together with some booster biocides, and we know they are highly toxic toward non-target organisms and can bioaccumulate in the food web chain, posing a significant threat to our coastal ecosystem. So although they are very important because they prevent organisms from attaching to marine structures such as boats, so they're very important for the shipping activities, for example. So they have also these huge threats. And oysters have been in the center of this problem because one of the first antifouling chemicals that was used in large scales in the boats and in our environment are TBTs. And early on, we saw that they started causing significant effects on oysters, so survival, growth, and reproduction problems. And so once we realized this contributed to the ban of TBT in several countries. And so this TBT was banned, but we still have a lot of antifouling compounds being currently used. And some studies have been showing that some of these compounds currently used, they can have either mitochondrial effects, some show some immunological effects, but usually they study these in different model organisms. And so to me, these compounds, the antifouling compounds, seemed like a perfect place to start my search for immunometabolic disruptor chemicals. And now you can see that I put a cell in the middle of this diagram of my project. This is because I believe that organism, pollution, and pathogen interactions, they start very, very small. They start at the cellular level. So it's imperative that we study these interactions at their roots, where they began. And for this, we need cells, we need a model. And so in my previous postdoc, still at Lema, I was working on the development of long-term hemocyte cultures. So hemocytes are the oyster's immune cells, so the cells circulating in the blood of bivalves. And yes, bivalves have blood. This is a question I get a lot, which is called hemolymph, but it's transparent. That's why, if you eat oysters and you don't notice the blood, it's transparent. So the immune cells are there, they're circulating and contributing to the protection of the organism. So I was able to develop these long-term hemocyte cultures and also different metabolic and immunometabolic biomarker of these cells. And so with this model, I'm able to apply a 3-tiered approach in the MAHIM project. So first, we will have some in vitro exposures to antifouling compounds. And as a first step, we will contribute to the hazard prediction of these antifouling compounds. So we'll look at their lethal effects. And with this, we will produce toxicological dose descriptors. So these toxicological dose descriptors, they give us an idea of how toxic a molecule is and their risk to living organisms. And it's a data that it's very important because in itself, it can inform regulatory agencies and help them identify chemicals that can cause effects at very low doses and apply a proper legislation to control their use. So this step is already very important, but what I'm most interested about is the sublethal effects of these antifouling compounds. And here is where I'll be looking at the immunometabolic effects of these compounds. But since it's a pioneer approach, we will also have to validate these in vitro findings in the whole organism, in vivo models. And so as a third step, we will have the validation of the in vitro predictions, and test if indeed these chemicals can cause disease susceptibility. So with this approach, I'm able to tackle my main research question, which is, can we identify immunometabolic disrupting chemicals, and can they be linked to increased disease susceptibility in marine organisms? I would like to highlight now some innovative aspects of this project. So we have an innovative methodological approach, which is the performance of in vitro toxicity testing under reSEArch-EU conditions. reSEArch-EU, what is this term? So reSEArch-EU stands for physiological oxygen levels. So in the air, we have about 21% oxygen, and as we intake this air, as we breathe, our cells, they will actually experience a much lower level of these oxygens. Depending on the cell type, they will experience as low as 1% oxygen in comparison to the 21% oxygen that is in air. And I was able to measure some of these levels in the oysters, and we can see that similar to humans, we also have much lower oxygen levels that the cells are experiencing inside the organs. So when we do in vitro research, it's important that we take these oxygen levels in consideration, because if we are exposing these cells to air, which is 21% oxygen, it's very, we have a hyperoxic stress to these cells. So one of the main hypotheses that I would like to address here is that if changing the oxygen levels in the hemocyte culture microenvironment can shift the cell tolerance to chemicals. So here we have a common dose response curve to chemicals, and so when we do these dose response challenges under reSEArch-EU condition, we can either make the cells more resistant to this chemical, so we need higher doses to elicit toxicity, because they are more in their microenvironment that they're used to, or not, or maybe this could lead to a higher susceptibility, because we know that stress can already upregulate some protective response that could also contribute to the protection against the chemicals. So we are underestimating the effects of these chemicals from what it was supposed to be in the real organism. So this is really setting the stage for a next level ecotoxicity risk assessment, because most people doing in vitro toxicity exposures, we are not taking this in consideration, because it's not so easy also to control the oxygen environment of these cells. But we see that it's very important. So this is a figure recently published in a review I wrote. And so we have these cells that once they are put in vitro, they are exposed to hyperoxia, and we know this causes a metabolic shift in these cells. So under hyperoxia, we can see that the oxygen consumption rate of these cells is lower. They can produce more reactive oxidant species, and also have some other cellular effects. And what does that mean? So for toxic exposure, so dicoid is an antifouling compound present in many marine paints, boat paints. And here we can see that actually the hemocytes that were challenged to dicoids under reSEArch-EU condition, they present more susceptibility to this compound than cells exposed under normal air oxygen conditions. And this is also true. This was a control I did with the pesticide rotenone, which we know causes metabolic effects. And so also here we can see that cells under more physiological oxygen conditions, they are more susceptible to these toxicants. And a very interesting result as well that I saw is that this antifoulic compound decoy inhibits oxygen consumption in these immune cells, in the hemocytes. And so now I would like to see if this metabolic change that this compound is doing can also affect the immunity of these cells, their immune capacity, and so may act as immunometabolic disrupting chemical. And a second innovative approach that the MAHIN project is bringing is that we are not only testing commercial antifouling compounds, but also green antifouling technology. This is due to our collaboration with the BioDimar platform. So BioDimar is a biotechnological core facility that aims to link marine ecology to marine biotechnology with a focus on identifying natural antifouling compounds. So with the collaboration of Claire Heliot, which is the head of BioDimar, we have at our disposal a library of potential green antifouling compounds to test with our model, with our oyster model here. So using this cross-disciplinary approach, the MAHIN project aims not only to identify toxic antifouling compounds, but also safe marine-derived antifouling compounds to support green chemistry and natural compounds used in the shipping industry. So this brings me to my final take-home messages. I really believe that tackling immunometabolism is crucial. It's a crucial approach to better understand the pathophysiology of marine diseases. And I believe that immunometabolic disrupting chemicals can be a major contributing factor to the disease susceptibility of marine organisms, thus threatening marine life and ecosystems. So it's imperative for us to identify which chemicals can be acting as IDCs, to control their use, and also it's important to provide safe alternatives, for example, using green technology to guarantee the sustainability of coastal ecosystems and our marine resources. And with that, I would like to thank all funding agencies and the teams that have been contributing to my work at LEMA, IPREME, and the university. Also some collaborators from the University of Montpellier in the IHPE group. So I have a lot of great people, helping me in this project. And yeah, I would like to thank everyone, and thank you all for being here today and listening to my talk.
Thank you very much, Danielle. Thank you very much for such an interesting talk, such an interesting research. Thank you so much. I do think that it takes into consideration many different, let's say, items or topics. You go from let's say, chemical pollution to analyzing new responses, to put up new methodologies for measuring responses. I do know that working with hemolymph from muscles is a difficult issue, and to give cells to do experiments and exposure to contaminants is not easy. And then you finished giving, let's say, new research alternatives for looking for natural products as antifouling. So we all know that TVTs, for example, they can produce a very strong, very significant, let's say, significant endocrine disruption in gastropods, for example. There are many different studies that corroborate this. The topic, the selection of pollutants, of chemicals, is very suitable, and this new approach, I do think is very suitable too. So now I would like to know if there is anyone that would like to make a question to Danielle. I think that's a great opportunity. If you want to find out something else about this interesting topic. Is there anyone? No, there is not. There is nobody. Charlotte Corpero. Good morning.
Yes. Hi, everyone. Yes, hi.
Afternoon.
I'm very happy to meet all of you. I'm sorry because I know very well Danielle, so it's maybe it's not the good way to ask her for a question, but in fact I have a question because I was hearing all the story. And Danielle, I am very convinced that the cellular level of immunometabolic change is very integrative of the environmental impact of pollutants and, for example, pathogens. And do you know if we have already some examples of long-term effects and profound effect on the physiology of animals if we change the immunometabolism? My question is really maybe not clear, but in fact, I can ask it again. If we change the immunometabolism because of pollutants, because of change in environment, do you think it can have a long-term effect, for example, on the progeny, etc.? Okay, well, this is the question. And is it already proved in some other species, maybe not bivalves, maybe not in other species?
Yeah, so really immunometabolism is now mostly a biomedical field. It's this term for ecotoxicological research. I have never heard anyone addressing this specifically. So as I said, many people are addressing the immune system, the metabolism, but not this integrative approach. So I cannot say much in terms of marine organisms, but what is known in invertebrates is that, but even in invertebrates, people don't explore that much the effects of environmental pollutants. So they explore that a lot in the context of diseases.
Yes, okay.
So, for example, they already know that immunometabolism is the core of, for example, type 2 diabetes diseases. You know, so diabetes is a metabolic and an immune disease. So they already saw that this mechanism is the core of these diseases. Okay. But yeah, even in the vertebrate field, not many people is studying immunometabolic disorders. So I really believe this is still a black box for us in most organisms that we study. But for example, we already know that there are many chemicals that cause mitochondrial damage. This is very well reported. And so if we have mitochondrial damage, and we know mitochondria is at the core of several immune pathways. So for example, there are viral pathways that they require mitochondria to be able to activate the antiviral response. And so if you have mitochondrial damage, you will inevitably impair these responses. And this can have chronic effects. So if you always have these chemical exposures impairing your mitochondria, once you need to respond effectively against a pathogen, a virus, for example, you may have a completely altered response, which will affect
Thanks a lot,
health and
Danielle.
disability.
Thanks a lot, Daniel. And I think if we can also imagine that if we have impact on mitochondria, maybe if it's a female, for example, I think it can maybe have, defects for the gametes, for example. And so maybe the progeny can be altered. I think it's a good, really good place to make science. Thanks a lot. Thanks a lot. Thanks a lot.
Thank you.
I can ask a question?
Yes, of course. The first yours. Thank you.
Thank you. Thank you very much, Daniela. Very nice talk. I have a question concerning the oxygen level in the different compartments of the oysters. It's very interesting that there is some difference between the heart and between the fluid and the tissue. Did you have some example of a species of bivalve of mollusk? Is it already the same thing that you observe, or is it different between the species? And also, I have done some oxygen level in different fluid, and in clams, for example, there is difference from the extrapallial fluid, the pallial fluid, and the hemolymph, completely different level. So very interesting for the immune response variation with oxygen level too. So my question is for other species.
Yes, so the actual oxygen levels within the fluids, early on, it was a little tricky to accurately measure because of some methodological limitations. So in the past, what some people would do, they would use hermetically closed, for example, needles, and they would extract the hemolymph and then use some machines to measure the dissolved oxygen in these fluids. But with this approach, you always have, you can have a little bit of oxygen contamination when you take this sample and then you put it on a machine. But even though with these limitations, there are a few studies that I found. They're not much in bivalves. So for example, there's a bivalve species in the Arctic that they measure oxygen, dissolved oxygen, their hemolymph. And also some in the oysters too, they have done this in the past, and we always see much lower levels than air. So in air we have 21%. Most of them they find between around 10-15% oxygen. But so what was a little innovative in my approach is because I had this real-time oxygen sensors, these probes that I was able to, they're very thin probe introduced in the animal while the animal is alive and breathing. And so it gave me real-time measurements of the dissolved oxygen with very, limited disturbing to the organism. And what I was able to describe that it was not described that is that it's also low. So I find between 5 and 10%, most commonly 5%, so lower than most people documented. And also as soon as the oyster closes, this levels drops to 0. So undetectable oxygen levels within minutes, less than 5 minutes. So the oysters goes from 5-10% to complete anoxia in a few minutes. So, and I really believe this can also affect the immune response. As we know, for example, there's a transcription factor which is very sensitive to oxygen levels, which is called the HIF1 transcription factor, hypoxia-inducing factor. And this transcription factor is also involved in immune responses, not only response to oxygen levels, but also immune response. And so, yes, I think everything is linked. And that's why, I really believe we have to bring this more integrative approach when studying such environmental effects and the effects of pollutants.
Thank you very much.
Thank you very much, Danielle. Is there anyone else that would like to make a question? I don't see anyone. Not anymore. Christina? Already did it? Okay. Yes. All right. For me, it has been very interesting all what deals with physioxia. I find it very interesting, and I was wondering if, for example, when performing neutral red retention time or alteration of the lysosome membrane using hemocytes, if we are underestimating the response, for example. We have done a lot of work regarding this biomarker, and to be honest, now I'm wondering if we are underestimating our results. So interesting point of view. And then when talking about immunometabolisms, and if we go more to immunotoxicity, I don't know if I'm just discussing, and I'm going to come with a question that maybe is not a framework, but I would like to know your point of view. Do you think that this alteration or adverse effect in the immune system, it comes more because of the chemical or because of the oxidative stress produced by the detoxification of the chemical? What is your point of view?
Yeah, this is a very good question. So because I can see more of like this integrative metabolism of the cell. I can see really many ways of how a chemical can affect immunity. And so one of them could be, as you said, once the chemical is metabolized, it can produce several reactive oxygen species. And actually, in this review that I mentioned that we published, the focus is actually on this, how redox reactions can control the physiology of the oyster and their immune system. And so we know that many immune-related pathways then can be regulated by proteins that are very sensitive to redox reactions. And so if you have excessive an oxidative environment in the cells, these pathways can be modulated through this mechanism. At the same time, we have, as I mentioned before, some chemicals that can disturb mitochondrial metabolism. And so these immune pathways can also be affected through this disruption of mitochondrial function, which can also increase ROS production, because if you have a dysfunctional mitochondria, the electrons won't go out through the electron transport chain and can end up reducing oxygen, producing annual superoxide, as many people working in that area know. And this is also a way that they can affect immune responses. So really, I find it fascinating to have this vision of how everything is connected and be able to provide this this approach when talking about chemical toxicity. Yeah.
Okay. Thank you very much. I think that that if there are no more questions, I would like just to thank you again for your presentation, for participating in this CU Talent. Thank you also to the attendees for their time. And as this is the last webinar of the reSEArch-EU project, I would like to very thank to Maria Yamcombaya and Beatriz Díaz Tarduño, that well, they are our technical managers, and they are behind all these organization issues. I mean, and without them, this it could not have been possible. And also to our section at the university, our marketing section, that is the one designing all our dissemination material. So thank you very much to them too. And to all of you, all the technical managers for the partner universities that have done their best to disseminate CU Talent and have looked for researchers to participate. I really hope that this one day help you to put in contact with the early stage researchers from the Alliance. Thank you very much, Danielle, and thank you very much, the University of Western Brittany, and thank you very much to all.
Thank you
Bye.
again. It was
Thank
a
you.
great opportunity and an honor to participate in this webinar. Thank you.
Thank you. Bye. Bye-bye.
Thank you. Bye.